Today, I'm going to talk about simulation-assisted discovery of membrane target nanomedicine. This work was done by my two PhD students, former PhD students, Dr. Huajian Gao, who graduated from Brown University with me and moved to Institute of High Performance Computing. So his picture is shown on the left. And my other PhD students, Dr. Vebleo, who recently graduated and he's a postdoc in University of Michigan. I actually been working at Brown, but I moved to Nanyang Technological University last year. This work is also in collaboration with a number of faculty collaborators. I list them here. So mostly I wanted to point out Dr. Wenpeng Zhu, and Dr. Woo-Sung Kim, and Dr. Manonakis, my former colleague at Brown University. Let me just share you a short story today about membrane target nanomedicine. I think it's very relevant to our current pandemic and other issues of urgent concern to society. Okay, I'll tell you what we do first. This is like summary slides. So what we're doing is the mechanics of membrane target nanomedicine. The scientific question we address is how to use nanoparticles, how to selectively rupture bacterial or viral membranes while leaving human and mammal membranes intact. So there we consider some nano-engineered agents, like some compounds, and we study their penetration, attachment penetration into the viral or bacterial membrane. After they penetrate into membrane, these compounds would aggregate, form so-called domain aggregations, and also cause deformation in the membrane. Then they will induce pore formation and fracture. The membrane, after you put this particle with the membrane, with a bacterial membrane or viral membrane, they would eventually fracture the membrane, such as shown in this video on the right. Okay, you can see we start with a vesicle, then we put this molecule in. They would cause aggregation. Let me play this video once more time. They would cause this aggregation, right? Domain aggregation on the membrane surface, and then eventually cause the whole thing to rupture. So we have a number of publications after today's lecture if you're interested. And also, let me just summarize. What my group does is we develop multi-scale simulation methods, including MD, coarse grain MD, and continuum mechanics to model a kind of a chemical fracture of cell membrane and help discover membrane target nanomedicine that selectively penetrate and rupture bacterial viral membranes. All right. So anyway, so the background of this, as I say, this is very much relevant to the current pandemic. While we know that there's a lot of virus pandemic over the last century, current COVID-19 is one of them. And let me just quote one sentence from Bill Gates. So he said, if anything kills over 10 million people in the next few decades, it's most likely to be a highly infectious virus rather than war. Right. Let me also point out there's another looming crisis of importance to human society, which is the antibiotic resistance. You know, antibiotic is a key pillar of the modern medicine. It has greatly extended the human lifespan. So the first antibiotic was discovered in 1940s, the penicillin, so in the 20th century. Since then, there's a war between new antibiotic, new drugs coming on market, and also the bacterial resistance. You know, as you use one kind of antibiotic for some years, the bacteria develop resistance. So the drug is no longer as effective as it first introduced. So the left panel shows increasing the emergence of drug-resistant bacteria. So in fact, we're facing a crisis now. According to this UN report released in 2019, the drug-resistant infectious disease could cause 10 million deaths globally per year by 2050 under the most alarming scenarios if no action is taken. So let me just very briefly say why we're studying membrane target nanomedicine. So I list a existing antiviral medicine, antibiotic medicine on the market on the right panel here. So most of this existing antiviral antibiotic medicine, they target some kind of protein, cell wall, or DNA synthesis transcription pathways. So therefore, they face some challenges related to mutation-resistant persisters. Okay, so you know, virus mutate very quickly, just like COVID-19. after it was emerges, at the beginning of this year or end of last year, has mutated tens of times. And also the bacteria develop resistance, right? The bacterial resistance develop, the main is because the so-called persisters, right? When you have a bacteria population, most of them are rapidly dividing, they're growing, so therefore they're easily killed by the existing antibiotic. But among this bacterial population, there's always a subpopulation. They're called slowly growing bacteria, and this bacteria is called persisters. Okay, so the persisters, because they're not metabolic and not as active, so they are very hard to kill by antibiotic, by the current antibiotic. So after you use the conventional antibiotic, this persister will survive and they develop resistance that way. So these challenges, as we summarize here, is most existing medicine on the market target is protein, cell wall, DNA synthesis pathways, right? They're effective against active bacteria, metabolic active ones, but they're facing challenges related to persisters or mutations, right? And the potential solution, potential mitigation strategy is can we develop a nanomedicine target cell membranes? Because membrane is a highly conserved component of bacterial virus, right? They undergo very slow evolution. So once you can selectively destroy membrane of a virus or bacteria, then it's very hard for them to get out of it, right? So this membrane target nanomedicine has the advantage of counter current challenge related to pathogen mutation resistant persisters. Second of all, can we use modeling simulation to help speed up the pace of drug innovation discovery, right? Because currently there's a rapid evolution of resistant bacteria and mutation. So can we use modeling simulation to help us speed up of the pace of discovery? Right, so let me just show you, to demonstrate these ideas, membrane target medicine, let me show you some quick examples. For example, we consider 3 molecules. One is adapalene, right? CD437, NTZ, DPA. These are chemical compounds similar in size, right? But it's a very tiny difference or minor difference. For example, the adapalene and CD437, the difference is only the root OH are different. NTZ, DPA, it kind of shows some differences in structures. So usually when we consider these compounds, we consider their dimensionality, whether they're particle, they're rod-like, they're plate-like, or some kind of shapes, or their polarity or their stiffness. All these can affect how they react with the cell membrane, right? For example, we can see that this experiment shows adapalene barely interact with cell membrane. So cell membrane are intact, right? They just kind of almost no influence on that. But if we expose the cell membrane, this is actually a vesicle, they're mimicking bacteria. The bacteria mimicking vesicle. If we expose CD437, which is tiny difference in its chemical root here, now you see a substantial change. Now this vesicle begin to show domain aggregation. These molecules, so this molecule would penetrate into the membrane and aggregate within the membrane. You can see this fluorescence in the membrane. And for this molecule, if we expose membrane to this one, it has even more severe reactions. It shows aggregation, and eventually the whole thing will burst, rupture, right? This shows you it's feasible to use these molecules to selectively penetrate in membrane and cause them to rupture. Right. So now we use modeling simulation to help us understand the mechanism and speed up discovery. So what we do is we use a so-called steered molecular dynamics. The steered molecular dynamics essentially allows us to compute a free energy evolution along a pre-selected reaction path. For example, in this case, we're interested in, as we bring the molecule from outside the lipid bilayer, the cell membrane, to the center of the membrane, how the free energy is evolving, right? So this is called potential mean force. So we divide this reaction path into many, many intermediate states, like 10 intermediate states. Around each state, we calculate, we sample the possible equilibrium states, we compute the free energy, right? So we essentially anchor this molecule using a spring, and cause their thermodynamic fluctuation we cause a sampling or energy. So we would obtain the potential mean force as a function of distance from bilayer center. The lipid bilayers from a center is 2 nanometer thick. So you can think of here is the surface of the membrane. So we're bringing a molecule from outside the membrane to the interior, right? So we encounter the surface somewhere here, then go into the interior of the membrane. So let me show you these 3 molecules I showed in the previous page. Adipolene, this molecule shows almost no interaction with the membrane. So we can use modeling simulation to confirm that. So as you bring this molecule toward the center of the lipid bilayer, you have an energy barrier, the energy goes up and then goes down a little bit. But even so, the energy within the cell membrane is higher than outside. So that means there is an energy barrier to bring this molecule to the interior. And it's energetically unfavorable to bring this molecule, which agree with our experiments. On the other hand, you can see CD437, if we modify the root a little bit, this blue curve, as you bring this molecule to the interior of the membrane, it shows a tiny energy barrier, and then there's a sink. There's energy sink within the membrane, right? So this energy is smaller than the energy outside. So that means it's energetically favorable to bring CD437 into the interior of the membrane, which in our previous page we showed the movie. They do cause aggregation. The NTZD-PA, that's the molecule that causing the vesicle to burst, right? It shows even more severe, this red curve. As you bring this molecule toward the bilayer, it shows the energy dip, right? There's tiny small barrier here, then there's even more stronger energy sink. So that means the membrane center has a strong attraction to this molecule, this red molecule. You want to suck it in, then that cause aggregation, cause all the reaction, aggregation and pore rupture. So this page shows very encouraging results. The simulated energy profile are perfectly consistent with experiments. It is predicted that adipolene cannot penetrate the cell membrane, and the other 2 molecules, CD437 and NTZD-PA, are capable of strongly penetrating the membrane. Based on this insight, we can go and talk to our collaborator from medical school, from chemical engineers. And then we actually consider several molecules, CD437, CD1530, aderotin, and adipolene, and we can map out their energy profile. So we predict, right? We predict the blue curve, these 2 molecules, the CD1530 and CD437, these 2 molecules are the feasible molecule to as the new membrane targeting antibiotic. Then we can use experiment to confirm that, and this shows with these 3 molecules, for example, adipolene, you increase in the concentration of this in the solution, and none of them went to the membrane, right? The fluorescence, the vertical axis is fluorescence intensity. With aderotin, with increasing concentration, some of them go into the membrane, but they have to be sufficient high concentration, as this the green curve show here. But with these 2 molecules, CD437, CD1530, these 2 molecules show up substantially within the interior of the bilayer. We can also use simulation to confirm this. Right, these 2 molecules we identified to be favorable as antibiotic. And this one, CD437, is the favorable one, adipolene is the unfavorable one. So we can confirm using simulation, indeed, this molecule can penetrate, insert into the membrane, while this one only attached to the surface without penetration. And these experiments help us to further verify the results. And while this one, the molecule penetrates into the membrane and cause aggregation, this one, this vesicle stay intact, so almost no interaction. Then our medical school colleague would further test this using the bacteria. So this shows this CD437 can kill the persisters, fast killing of persisters, right? When we increase in the concentration of this molecule, it can kill the persistence fairly quickly. And also we encounter very low resistance. So this molecule, ciprofloxacin, is the antibiotic on the market. It shows within 2 weeks, it shows substantial resistance, bacterial resistance. So as you use this drug for more than 2 weeks, you have to keep increasing their concentration for it to be effective. In comparison, the CD437 shows, and also this red one, CD1530, shows almost no resistance over 100 days. So this confirms our theoretical expectations, anticipations. So the free energy mapping captures the trace of membrane active compounds, and these MTAs show fast killing on bacterial persistence and low resistance. Now, a key question here, they have to be selective. So what is the basis for selectivity? Why do they leave the human cells intact or animal cell intact? Why do they only kill bacteria? So here we demonstrate. So here we use the bacteria mimicking bilayer versus a human mimicking bilayer. In a human cell, there are lipids, there are cholesterols. The human cell, the cell membrane have 15% of cholesterols, which substantially increase their density, right? So when we bring this molecule to the interior, in this case, we consider Pacino. Pacino is an existing drug in the market for antiparasitic, which we find can also be used as a membrane targeting antibiotic. When you bring this Pacino to the interior of the membrane, and the blue one, the blue curve shows bacteria mimicking membrane. So the Pacino can easily penetrate into the bacterial membrane with no energy barrier. But with a human mimicking membrane, it shows a very high energy barrier because of the presence of cholesterols actually become energetically unfavorable to bring this molecule inside. So we can, using simulation, to confirm this is indeed the case. Right, with a human membrane with the cholesterols, they present sufficient resistance for the penetration, but for the bacteria mimicking membrane, eventually this molecule would penetrate into the membrane. Okay, well, you see this movies. We also have conducted experiments to verify with a human membrane, right? Increasing concentration shows no fluorescence. And here shows with bacteria membranes, with increasing concentration, then these molecules do penetrate into the bacteria. Now, this one shows for the, so this molecule can kill the persisters quickly and shows almost no toxicity to human cell. All right. Now, in the interest of time, I don't have too much time to talk about the mechanism and the modeling and so forth. Let me just mention briefly, this is multi-scale modeling. I've shown you some atomistic scale, atomic scale, energy mapping and simulation, but there's a lot to be done under a continuum, under a larger scale. For example, we can model the domain aggregation and pore formation by the fracture process. So we write down the free energy that consists of several terms. One is the bending energy of the membrane within domain and along the outside domain on the membrane, and also the line tension energies of the interface along the domain lipid interface and the pore boundaries, and plus remote tension, right? The remote tension. Then we can do the energy variations to model the whole process. We can identify the driving forces for the pore formation, which are the bending energies, because this molecule, after penetration, they sit asymmetrically. They cause membrane to bend, right? So the bending energy, interfacial tension energy, they are primary driving force for the pore formation rupture. Right? In interest time, I won't go into too much into the details. And with the viruses and the selectivity for the antibiotic, these are cholesterol-based selectivity. For viruses, we use a size selectivity. Okay. As you know, the human cell, mammalian cell, these are typically 10 to 100 micrometers large, and bacterial cell, they're typically a few micrometer, but virus is very small. Okay, we're dealing with antiviral, and then you're dealing with about 100 nanometer size. These are viruses. So my colleague, Professor Nanjun Cho at NTU, have developed these antiviral peptides, as shown here, which exhibit a strong size-based selectivity. For example, when we expose different liposome of different sizes, and these antiviral peptides can rupture this liposome selected based on their size. If they're below 150 nanometer, they're ruptured very efficiently. The larger they are, the less efficient these peptides become. This is called size-based selectivity. Then we can test these antiviral peptides on the broad spectrum of viruses, Zika virus, dengue virus, Zika, and the other viruses. So they're all very effective. There's a broad spectrum of efficacy against all these Zika, dengue, yellow fever, JEV, and chikungunya viruses. Now my colleagues are testing this actually on the COVID-19 at this moment. And yet they show very minor toxicity to human cell. For example, mammalian cell, the viability shows that you need a huge concentration, right? These are 10 micromole concentration to have any effect on human cell. While we have tiny concentration, they start killing these viruses. And we think this idea, the membrane target nanomedicine, can also maybe can use for cancer therapy. For example, cancer cells also exhibiting some kind of difference from a healthy cell. One example is this lipid called phosphatidylethylamine. Right? This PE, or short as PE, for healthy cell, the PE only existing on the lower leaflet of the bilayer. But for cancer cell, and this lipids show up on both outer leaflet and inner leaflet. So this presents a target for membrane target nanomedicine, right? So far, there's in the literature, people using this peptide to cause the pore formation. And so this is another, along similar ideas, we can develop nanomedicine to target cancer cell, target viruses, target bacteria. Right. So over the long run, we aim to develop a simulation-assisted platform for discovery nanomedicine. Current platform for discovery, they're based on screening of chemical space, right? You establish a big a drug library through combinatorial chemistry. You basically synthesize large number of compounds, then you test them using the C. elegans, the worm, right? The worm kind of assays. The C. elegans survival screen. You grow these worms from larvae to adults. Then you put them in many of these wells. Each well you put in some drug. You put in different compounds as potential drug. Then you culture them for 11 days. You see which wells the bacteria have been killed. Right? Sorry, you also infect them, right? Which of them has been killed, which of them survive. And this will allow you to screen which compound or potentially can be used as antibiotic or antiviral, right? So this current platform, then after you succeeded here, you can study biomimetic vesicle to study mechanism. Then you can further transfer them to cell-based assay to confirm cell permeability, anti-persister activity, and cytotoxicity, bacterial activity. After you've gone through this process, you can go to animal-based assay and clinical trial. This is a long process, taking years. Right? And this process here, for the screening case, this is pretty expensive and time-consuming, low hit rate and inefficient. They're hard to improve, and also the mechanism is hard to identify. But today, using the multi-scale simulation methods, we're hoping that over the long term, we can supplement or replace this screening step using modeling. Okay, we can use modeling to model attachment penetration of this molecule into the lipid bilayer. We can start aggregation, pore formation, and establish a machine learning algorithm to train the machine learning to help us screen this fast and low cost. Right. So perhaps the modern simulation may accelerate the initial screening, assessment, design, optimization, membrane target nanomedicine, and reduce cost. Let me conclude my talk right today. most existing antiviral, antibiotic medicine target some kind of protein, cell wall, DNA, RNA transcription pathways. These are usually effective on metabolically active pathogens, but they're less effective for persisters, for slowly growing bacteria, or if the virus for virus mutation, they also become less effective. So in contrast, we think membrane target nanomedicine offers advantages in countering challenges related to pathogen mutation resistant persisters. We have been trying to develop multi-scale modeling simulation methods to assist the innovation discovery of this medicine. Over the long term, this research, we aim to establish simulation-assisted drug discovery platform for accelerated drug screening at reduced cost. So with this, let me end my talk. Thank you very much.